This is a production of Cornell University.
I'm going to talk a little fast because there's a lot of stuff I want to go over. I'm from the state of Minnesota, crazy place, the only place to elect a professional wrestler as governor. As governor, not many places do that. Some things about Minnesota: second largest variation in temperature in the continental U.S., 38°C to minus 53°C. 90,000 miles of shoreline, more than California, Florida, Hawaii combined. We measure every puddle. The largest population of people of Scandinavian, Somali, and Hmong descent outside of those countries. So we are somewhat diverse. I'm at the great University of Minnesota, 55,000 undergraduates. It's a big school, home of Norman Borlaug, who won the Nobel Peace Prize, Honeycrisp apples from Jim Luby in my department. Something happened. Oh. And where they discovered the pacemaker, which is that strange machine down on the bottom, Medtronic spun off of. Largest city is the city of Minneapolis, Minnesota, a couple million people. And Minneapolis has a park board. I'm chair of the park board. The park board existed before the city of Minneapolis. And so I help run a park system with 183 parks and 50 recreation centers, 1,800 kids sports teams. I have my own police force, which is something. 6,732 acres of land and natural area that we manage, and then there's a bunch of other statistics. I'm not going to talk about that now, but I do know there are some people who are interested in urban agriculture. I'm instead going to talk about plants and stresses in plants. Now, some changes that have happened since the 60s. Why did I even get into this area? The crops we grow in the 60s, which is the last time a lot of work in CO2, photosynthesis, and some of that work in floriculture was done, it's changed. The industry was cut flowers and potted plants. Now the industry is based on bedding plants, and the number of species we grow has increased dramatically. Energy costs have increased, and profit margins have decreased dramatically in the industry. We now have HPS and LED lighting, which was not as commonplace back then, and we have shade curtains and environmental control computers, but we don't have information to put in them. And temperature and CO2 levels have increased. When I was in graduate school with Marvin, the standard was 333 parts per million. Right now it's 388. So there is little to no information on lighting, temperature, and CO2 standards for the current crops we grow. Even the geraniums we grow now are different than the geraniums that were grown in the 60s and 70s. And we don't have any information to utilize this fantastic equipment that we have that run our greenhouses. I also want to talk about a little bit how we stress our plants out on a regular basis and often don't appreciate it. We have cool season crops and warm season crops, and this is a relative growth rate curve of two kinds of turf. But notice with cool season crops, and I'll show you data to this effect, 20 degrees there's a maximum C, and notice above 20 degrees you actually start having a decline. And I'll start showing that with photosynthesis and development rate on some common crops we grow. I think we stress our plants out commonly and have no appreciation that we are in fact doing that. And I'll show some evidence of that. I'm going to talk about abiotic stresses. As Neil mentioned, I'm going to talk about light, temperature, and CO2. I'm going to talk a little bit about biotic stresses, and then I work in a variety of other areas, and I just want to give you a little bit of an idea what some of those are. This is some data of the average daily light integral, or moles of light per day in January throughout the United States, and you can see that you, as Minnesota, is at 2 to 4, and at most 6 moles of light, and that there are parts of the United States that benefit greatly from adding supplemental lighting, and you can see that Cornell is in there as well as Minnesota. Why do we light? Well, when we do light, there's some benefits. Here we have a graph of daily light integral, and this is cutting yield per pot, and this is for taking cuttings off of a pot, stock plants, and we can see that as the daily light integral goes up, the cutting yield goes up, and we can also see with two different levels of CO2, the yield goes up. But in generally, yield of any crop, be it tomatoes, greenhouse peppers, lettuce, dry weight, is proportionally increases with photosynthesis. This is actually a picture from Neil, I think, isn't it, Neil? You recognize that picture. Neil took this picture a long time ago. This is a purple wave petunia with night interruption lighting, and this is supplemented with 150 micromole of additional high HPS light, and this is just to demonstrate both of these have long days, but which one flowers quicker, the one with the higher light level, or PAR. It's a standard photosynthesis curve that's been used forever and a day. It basically shows that as absorbed light, or PAR, goes up, CO2 assimilation goes up. There is some region that's typically in the winter in our area where plants are light limited, but once you get up to a certain level, which is typically around 400 to 600 micromole, saturation occurs and the plant photosynthesis is CO2 limited. So we started collecting data. We use a light core 6400. We did 25 different species, so we have huge data sets. I'm just going to show you some brief ones to make some points. This is impatiens. You can see that impatiens, as PAR goes up, PN goes up until you reach approximately 400 micromole of light. Okay. Is that a single leaf or canopy? No, you'll see some standard deviations. So what we do is we take 5 different plants. So each one it's 5 separate curves from 5 separate leaves. And then that's replicated again 3 times over time.
Horizontal leaves or the plant, whole plant?
One horizontal leaf. So we use a light core 6400, which clamps right on. Yep. And I'll come back to something related to that question a little bit. This is from a tomato grower. This is Bushel Boy tomatoes. Each of these represents a different cultivar. I just want to show that within a species, even within a single greenhouse, you can have very different rates of photosynthesis, and you can have saturation occurring at different spots. This is 750 micromole, so it's higher than the impatiens. And notice that this particular cultivar was still increasing. So there's dramatically different responses to increasing light, suggesting that even within the same greenhouse, there should be different light management strategies based on the cultivar that's used. In addition, we looked at some leaf filtering, and this has not been considered really, but here's coleus and here's panicum, a grass and a coleus, and then we just measured light intensity as you move down through the canopy, showing that you have significant filtering. Now these plants were just barely touching, okay? But this just goes to show you that only 60% of the incident light in a coleus canopy actually gets to the middle of the plant, and by the time you get to the bottom of the plant, you're down to 18% of radiance that actually can penetrate. In contrast, of course, with the grass, it's better, 80% and 55%. And then we shade. We have these shade curtain systems. You have them in your greenhouse, but how do we decide when to close the shade? Who decides? Bill, who decides? You divine it? Yep. We divine it. There's no information with which to use to identify when we do this. Now, I'll tell you that most greenhouses use a standard of 3,000 to 4,000 foot candles, which is comparable to around 600 to 800 micromole, and then the shade system starts closing. Also, in our systems that we have, if you go out to your greenhouses, the light sensors that control the shade systems, where are they? They're on a weather station that's on outside of the greenhouse. Okay? In general, only 60% of incident light enters the greenhouse. You have more than that, because you have single pane glass in a lot of your greenhouses. Most places have X-light or double poly or another kind of poly that diffuses the light, so yours is actually better. And the shade cloth systems, typically the standard shade cloth is 55% light reduction curtain that comes across. Therefore, if you use a typical shade system with 55%, when it closes at 600 micromole of light, okay, the radiance of the top of the canopy after getting into the greenhouse and going through a shade system is all the way reduced to 198 micromoles. And that's at the top of the plant. And if you go to the middle of the plant, in a typical coleus canopy, you're down to all the way down to a third of the actual light that comes in. To look at an impatiens curve at 600, this is where you're at, getting into the greenhouse and the shade cloth system closed. And then this is halfway down on the canopy, down to 119. You've reduced your photosynthesis in that particular instance from close to 7, okay, all the way down to just above 2, a third of the photosynthesis. You've effectively starved your plant at that point. And greenhouses are routinely doing this, that they're overshading all the time, which is creating significant problems that they're not aware of. So for instance, in a cut rose greenhouse or a greenhouse tomato facility, they have more blind shoots in those facilities with roses, and they have less fertile flowers with tomatoes. And the whole problem was because of the shade system, because they weren't using good numbers on the shade system. The solution, first of all, is quite simple. Take the meter that runs the system, take it off the top of the pole outside of the greenhouse, and move it into the greenhouse, into the middle of the canopy where the plants are growing. It's not too complicated, okay? It's very simple and straightforward.
Why not simply reset the figure?
Reset the figure? You can, but the problem is this, that during, we're in the north, and the sun comes up, and up here, it comes up, in Minneapolis, it gets up to 23 degrees above the horizon on Christmas, and then goes back down again. When the light barely comes up like that, more skips off the glass. And so still, so in other words, what gets in the greenhouse changes depending on the sun angle. So simply the best thing is just put it in the greenhouse, which none of the greenhouse companies do, and which actually absolutely would be the best thing for all of you to do, and then run the shade system off that. That works for any greenhouse. I don't know of a single greenhouse in the United States that has the light sensor where the plants are. Okay? Switching topics to temperature. The world is a warm place, and it's getting warmer. Okay? It's a premise that I work off of. I work in a lot of heat stress. We are routinely causing heat stress in our crops, and most of us don't appreciate that. Now, plants naturally try to do things. They'll reflect light as best they can in warm places. But we often will stress many of our crops. Bushel white tomatoes, which is a large greenhouse tomato producer, their lowest yield is the summer, which is not what you would expect, but that's when yield is the lowest. Why is it the lowest? When we started this research project because of them, it's simply because of heat stress that occurs on tomatoes. Okay, species differ in the optimal temperature for photosynthesis, and that optimal temperature can actually change as the season progresses. Okay? It's my belief that we're routinely reducing photosynthesis by growing plants too warm. I'll show you some examples. Here's a curve. This is pansy. Temperature C, there's 3 light levels, 300, 600, and 900 micromoles. And here's PN, and you can see the pansy PN is optimal at 20 C. Okay? You can also see that as you go above 20 degrees C, which the greenhouses probably are even today, that photosynthesis starts to decline. And you can see this on the plants. Here's 2 plants, and sorry for it's in Fahrenheit, but there's constant 20 C on the left, and you have 30 C on the right. You can pretty clearly see the 30 C pansy has less photosynthesis that's occurred with that crop. There's a group of crops that I would say are mid-range. This is gerbera, and you can see it is an optimal temperature between 20 and 25. And then you can see a shift still further with poinsettia, where you'll have an optimal temperature with poinsettia around 30 degrees C. And this is why we can raise poinsettia stock plants in a greenhouse during the summer. But a question we started asking is why, or what is the justification or cause for these responses, for these curves to shift? Because the photochemistry should be essentially the same. We started looking at the stomates and just plain old stomatal conductance and stomatal behavior. And if you remember the pansy plot, this is actually temperature versus stomatal conductance. You can see it's quite high, and as temperature increased, the stomates actually closed. The limitation that occurred with photosynthesis may be associated with closure of stomates. Okay? If we look at gerbera, and look at the stomatal conductance with gerbera, we can see that we also had, here was around where we had the optimal temperature, and then you can see the decline again. Suggesting that, frankly, if the stomates close, of course the plant is going to have less photosynthesis. And that may be part of the justification for that. Something else we looked at is short-term high temperature. Okay? What we found was that a short-term, and by what short-term? 2 hours. Okay? Now we focus on temperatures that are not temperatures that people would call heat shock. There's lots of research on heat shock, and that typically is at temperatures of 40 to 45 C. We're looking at temperatures that are 30 to 40 that plants see on a very common basis in a greenhouse. But we asked the question, if we let the temperatures heat up to 95 degrees Fahrenheit for 2 hours, was there any impact on a plant? And there was. This is a graph of PAR on the bottom versus PN, and you can see a typical curve right here. But look at what happened if the plants were exposed the 2 hours at 35 C, and we redid the curve. We had a fully a 59% reduction in the maximum photosynthesis the plant could do. We went back in the next day and measured again. We were still depressed. The next day, it came up. It took 3 days for the plant to recover from that single 95-degree exposure. We worked on some other crops. This is petunia. This is glow lavender shades. We saw the same response. Significant depression and 3 days to come back up. So these short-term high temperature exposures can have a long, a 3-day or more effect. Growers are in fact doing this. Why? Because they want to save money. They let their greenhouse temperatures increase at the end of the day to capture heat and keep the greenhouse closed, hoping that they'll keep the heat moving into the night. Now, the Israelis have done some work with this actually as well. They did work with field tomatoes, and part of they saw the midday depression in photosynthesis that often occurs. And so what they did is they irrigated the field tomatoes and cooled the leaves when the leaves got really hot at that one time of the day. And they found they got a significant increase in yield as a result of that, and it eliminated the midday depression of photosynthesis that occurred from that high temperature. I wonder if we could apply some of these concepts in greenhouses as well, or greenhouse tomato production. Essentially, any time the temperature exceeds the maximum, you should do whatever you can to try to reduce the temperature to the optimal temperature range. I'll mention one other thing. We called around and we started looking for what are there fundamental differences between heat-tolerant plants with respect to flowering and heat-intolerant. So what we did is we called companies that produce flowers, bedding plants of different kinds. Send us a geranium that's very heat-tolerant and send us a geranium that's very heat-intolerant. And then we did photosynthetic response curves. And in every case, and these are across companies and across species, in every case, the heat-tolerant plant, which is in red, always had a higher photosynthetic rate than the heat-intolerant plant, suggesting that there may be some association with heat tolerance as well. And it brings to mind, we did some work on some Arabidopsis as well. Everyone works on Arabidopsis at one point or another. And what we did is we looked at how different kinds of heat stresses, we did 1 hour, 2 hour, 4 hour, 8 hour, 16 hour, how these different stresses at different temperatures. So we imposed all different kinds of stress because the term stress is a difficult term to define. Because how do we define it? What is it? Is it 8 hours at 28 degrees or 1 hour at 36 degrees? And what we found is a very strange relationship, and we published this back in 2005, and we looked at we evaluated a number of different Arabidopsis strains. But what we did is we put it onto a degree day model, and the data fit almost too close. It's one of these situations where the graduate student comes back and gives you something, and you think they had to have moved points. But you can see these points fit the models, and what this was the data suggested there was a cumulative effect of these different stresses, okay, with a degree hour concept over 32 degrees, okay? And that, for whatever reason, when we exceeded 225 degree hours, okay, over 32 degrees, you could see that we went from no abortion to completely aborting all the flowers on Arabidopsis. We don't understand this, but it didn't matter how the stresses were arranged. In other words, a 38 degree stress for 3 hours or 34 degrees for more time. It was the cumulative effect in degree hours over 32 degrees. So taken together, if you look at the stomatal conductance stuff we have, it brings to mind a couple questions. Do some plants either produce more ABA, okay, those plants that were heat sensitive, Do they produce more ABA in response to a chronic high temperature stress? Or are they more responsive to the high temperature stress, to the same endogenous ABA level? Because the responses we're seeing time and time again are related to stomatal closure, not any damage to the photosystem in the temperature range I'm talking about. Okay? Okay. Can stomatal conductance, after a single stress, be a way that we can screen for heat tolerance? Because after a heat stress, even a 2-hour heat stress, if the stomates are staying open and the plant is still actively photosynthesizing, it likely is going to be more heat tolerant than one that is not. So this is a screening protocol that can be used. CO2. We went back and started looking at CO2. All the greenhouses in the United States, people who've been around for a while remember, there used to be CO2 burners in greenhouses. We got away from them because the burners would not work properly and they'd release ethylene. You'd have disasters, and so people got away from using CO2 burners. So if you go around to greenhouses now in floriculture, there's next to no CO2 being used. Here's cyclamen. Down here is ambient. All the way down here. Look at the degree of responsiveness that you can get on cyclamen by increasing CO2. Here's greenhouse tomatoes. Now this particular grower is, the greenhouse tomato growers and greenhouse vegetable growers do inject CO2, typically up to 800 parts per million. So you can see that's here, and some of these species still would benefit. But the bottom line is there was huge, huge responses to CO2, more than what we appreciate. We went into the research greenhouses and looked at what are our CO2 levels on a sunny day in January. Here's the meter reading, 193 parts per million. 193 parts per million. Where did the CO2 go? The plants used it up. The new greenhouses we have, we've spent so much effort and time making them tight that the CO2 levels in the greenhouses are actually significantly depleted. Significantly depleted, and we don't even really appreciate this. To give you an idea, this is 1 hour under what we consider low light, medium light, and high light. Started at 388, 1 hour in a low light greenhouse, the CO2 level dropped to 370, 1 hour in medium light down to 340, 1 hour in high light all the way down to 310 in 1 hour, in a single hour. Okay, now I can tell you if you have all kinds of high pressure sodium lights up there, after 2-3 hours, your plants, if they're down to 190 parts per million, they're not using all that light. You're simply wasting the light. The new open roof greenhouses that people recognize gives you superior growth in the industry. Part of the reason I believe is, frankly, that these have CO2 levels that are similar to ambient CO2, and they're not CO2 depleted. The addition of CO2 is the #1 most cost-effective way to actually increase photosynthesis in a greenhouse today. Not all the lighting. It's the #1. If I take a CO2 sensor and go into a canopy on a bench, on any of your canopies, okay, you'll check and you'll find that your CO2 level inside that canopy is often around 200 to 300 parts per million. Okay, even when the greenhouse is venting, because you get this boundary layer that forms on the bench itself. In some cases, doing nothing more than circulating your air at least equalizes the CO2 level within the greenhouse, so you don't have depletion within a canopy. At least you'll get up to 380 if you're bringing in air from outside. And I think in our experimental greenhouses and in our growth chambers, we are commonly depleting or growing plants in depleted CO2 levels that are actually dramatically affecting our results, and in some cases invalidating them because the plant may not even be able to use the light that you're providing in the growth chamber itself. Switch gears a little bit with succulents. There's a lot of interest in succulents. This is just to show. This is a crown of thorns, and succulents respond as other plants to increasing CO2. This is during the day. Now there are plants, this is Echeveria and Aeonium, these take up CO2 at night. Not all what we consider to be cam plants actually do take up CO2 at night. We studied a number of cacti, and they're taking up CO2 during the day in many cases. The assumption is that they're only taking CO2 up at night, and that's not the case. Also, orchids, Phalaenopsis orchids, will take up CO2 during the day. But this is phragmipedium, and this is the beginning of the night, and this is CO2 uptake, and you can see pretty clearly that this orchid takes up CO2 at night only, suggesting if you're producing this orchid that you should actually be injecting CO2 at night. So there's differences. I'm going to move along quick. High temperature slowing of development rate. Leaf unfolding rate, this is an old graph I've used from African violets, follows this standard line. So this is a pretty common curve for lots of plants we have. We have an optimal temperature, and frankly, leaf unfolding rate often is optimized right around 85 degrees Celsius. And this was developed, here's African violet. You can see leaf number increasing up to here, and then you can see a decrease. And this is what I want to focus on. Because we have decreases, this, for whatever reason, this temperature is almost sentinel to lots of things. This is melon fly egg development. And doesn't it look an awful lot like the graph we just saw? But my point is this: when the temperature exceeds the optima, which in greenhouse production it commonly does, we actually slow the rate of leaf unfolding and slow the crop down in its development rate. Okay? These hot temperatures, and right now, your leaf temperatures, remember one thing, if it's 85 degrees in the greenhouse, your leaf temperature is above 90 and approaching 95. You are actually on the other end of that curve, and your crop is actually starting to slow. It'll take longer to flower. It's an underappreciated area. Using the standard curve, if we go from 85 to 95, that same plant to unfold one leaf will now take 5 days instead of 3.3. You've slowed leaf unfolding approximately by a third. So to come back again, we have a number of crops that are down in this area, and we have temperatures that are routinely going from 20 up to 35 C, and we are routinely decreasing photosynthesis as part of that process, and we're routinely decreasing the leaf unfolding rate and actually slowing the crop development in that process. And we're routinely closing our stomata. And someone was talking about edge burn on lettuce. Well, the high temperatures encouraging edge burn on lettuce. You can kind of understand where that comes from. If the stomates are closing, of course the plant is going to have difficulty taking up calcium in that scenario. Now we've gone through all different kinds of crops. I'm not going to go through this, and identified what happens if you decrease temperatures and the timing of that, and then we've integrated that into a package that everyone has access to. And if any of you haven't tried this program, it's a wonderful program. It's called Virtual Grower. It's free for everyone. USDA has it. It's used for modeling crop growth and temperature. You can literally take your greenhouse and identify how much more it's going to cost if you switch the outer covering on the greenhouse or not. So that's how we pull it together. The last thing is biotic stresses. I think most of us appreciate that when a plant is attacked, it releases volatiles that go to other plants, sometimes within the same species, sometimes other species, and tell that plant that it's being attacked. Plants communicate via volatiles. There's a plant, this is from data that is not mine, from 2001. This is showing ulnus that's uninfested, and here's an ulnus that is infested. And here's the GC depicting the dramatic expression and variety of volatiles that are produced after infestation. This is also a recent picture just showing you the degree to the sophistication. This is a recent paper. So the insect lays an egg. This is on Arabidopsis. That egg actually produces a compound that stimulates salicylic acid, which is antagonistic to jasmonic acid. Okay? So this actually creates a system where the plant is not able to defend itself. So when the larvae does hatch, the plant doesn't know it's being attacked and is defenseless. Started looking at what happens if we expose plants to methyl jasmonate, one of the primary volatiles that plants give off to warn that an attack has been made. What we did is we would treat plants, okay, with methyl jasmonate, and then we would infest them with mites. Okay? And this is how many mites fled the plants. Okay? So you'll see that in each case, a plant that was exposed to methyl jasmonate, okay, and this was for a single hour, resulted in a plant that when you would put mites on it, the mites would flee, okay, compared to the control. Also, if we quantified, if we come back to the tomato, you'll notice the tomato had significant effect. If we look at, we quantify the protease inhibitors, which is one of the purported responses that plants have in response to methyl jasmonate, you'll see with tomato that as methyl jasmonate concentration went up or exposure went up, the plant produced more protease inhibitor in response to that. If we take that further, this is the number of mites that were on the plant, okay, after a period of 3 weeks. And you can see that in any case, whenever there was a plant that was exposed to methyl jasmonate, it had fewer mites. That was the same case for aphids. Notice the control, we had 22. This is after 11 days with aphids. 22 aphids, we had methyl salicylate, but notice methyl jasmonate, we had a fraction of that. We had 8 in that case. This is just a representation of some of the work that we have. But clearly, in all the studies we did after a plant was exposed to methyl jasmonate, it actually, not only did the plants or insects try to flee it, but they actually proliferated less. Now, predation of any kind and activation of plant defenses has a cost for plants. And so actually, when you turn on plant defenses, that plant will actually have less dry weight than a plant adjacent to it if the defenses are turned on. The plant is spending a lot of effort actually producing compounds to defend itself at the expense of other materials. So there's a cost to this. And something that's also very interesting is that if you look at different kinds of wounding responses, you'll only see response, okay, to a caterpillar, not a tearing, saying that somehow the plant recognizes a pest. I don't know why this is blurred on this one, okay? But the bottom line is FAC is actually caterpillar spit, okay? The plant actually perceives the saliva from the caterpillar, and that's what causes the chain reaction and response. It's very, very specific. So we're in the process right now of actually determining whether plants have vocabulary, because I think that the response to a whitefly may be different than an aphid, which may be different than a mite. So we're looking at those areas. I will be very brief. New directions. We're doing a lot of new crop work. This is with South Africa. This gives you an idea of some of the gladiolas that exist in South Africa. We have very little gladiola species in the United States. We're doing work with cactus and succulents where we think that they can be new bedding plants or new potted plants, and we've been identifying plants and protocols to get cacti and succulents to flower when we want them to, so you can have them blooming all year. Doing the same with succulents of different kinds. Then we identify protocols. You can pretty clearly see that when photoperiod exceeds 12 hours, with all those species, these are each kalanchoes, that they remain vegetative, that if the photoperiod is less than 13 hours, they're reproductive. We've done a large project on green roofs in the Midwest because the green roof work that's been done in Baltimore and the East Coast and Chicago and Toronto doesn't apply to the Midwest. So we screened 78 species over a series of years and identified what plants will survive on a green roof in the Midwest. It's a crazy project on grafted cactus. Okay. Someone say there aren't many grafted cactus. You're wrong. There's a lot of grafted cactus in the world. Okay. Each of these cacti is on a different rootstock. It's the same scion. So the effect you see is purely a rootstock response. There's a lot of potential for this as far as expanding the crop palette. There is some interest in aquaponics. I will just mention a couple things. It's a difficult area. We talked this morning about how you have to optimize. You have to optimize the fish number that will survive. You have to optimize how many plants a fish population can support. And then as the plants grow, the conditions that you have changes. And the market, you know, people are getting tired of tilapia. There's only so much tilapia you can eat, right? Okay. So, and we think, like we have the Schlitz Brewery is converting into a giant aquaculture facility in Minneapolis. They're going to be producing more tilapia than we know what to do with. The interest in our growers is frankly in alternative aquaponic crops. And so what's an alternative crop? This is what's called a red claw, which it has no red claw, it has a blue claw. I don't know why they call it red claw. But it's a large freshwater lobster. It's actually a crayfish that gets a really big tail. It's from Thailand. Shrimp, crabs, and freshwater oysters. There's a lot more interest in this, and the restaurants in the Minneapolis area are very interested in this. And there are some that have some of these crazy systems where there are fish at the bottom. This is LEDs in the center. Okay, and this is lettuce in this case, but actually they've switched to basil, and this rotates. Okay, okay. My question was, why? Why not just grow basil in the greenhouse someplace? It'd be a lot cheaper. But they effectively do this. And so it works. And the last picture is this contraption, which is actually in California from 2 weeks ago. And someone has to identify protocols for these guys. This is Tom and Jerry. And so, so Tom and Jerry work, and they see each other, so they don't ever smash into each other. And what they're doing is they're spacing plants. So those guys will work for 6 hours, okay? And then you put a new battery pack in, and then they keep going. There's no overtime. There's no health care. There's no complaints. But someone has to program this thing about what a plant looks like and what to do. And there's actually a project we're looking at. This is these little helicopters with a little tank of pesticide. And the helicopters will fly. They will recognize an aphid and then spritz it. And the intent is to use it in orchards so that these things will target the shoot tips. So in Minnesota, none of the water flows. It all just sits like this in these big mosquito pools. But this is land I have. But anyway, I'll answer any questions you may have. I was supposed to get done by 1, and I am done 20 seconds early. Yes?
In some of your early slides, you showed you showed stomatal conductance data. The LICOR also calculated CI, internal CO2. What did that look like, and are stomates actually optimizing their conductance to maintain a constant CI so that they're not...
That's a good question. That's a good question. We didn't find that to be the case.
No.
We didn't find that to be the case. And the other thing I wonder about is that you know that as you increase CO2, let's say in a tomato greenhouse or something, the stomates actually close down some. They get smaller to accommodate some of what you're talking about. But I don't know what impact that has on actual photosynthesis. I still believe that there's a decrease. And the short-term heat stress we saw, that lag, seems to support that. There's some sort of response that a plant has, that some of these plants are responding to something, and I assume it's ABA. So the next step would be actually to look at some mutants, ABA mutants, to look and see what sort of response happens within those mutants themselves. But as far as internal CI, we didn't find the optimization or tried to get a constant internal CI. That was not the case. But yes, we use the LICOR. Yes?
Could the differences in the internal CI explain why you were seeing different light response curves in the same? Which could the differences in the internal CI be explaining why you were getting different light response curves, where you had power versus PM? For the same
You mean within
variety,
the same species?
Yeah, within the same species, different varieties. How would you explain
I don't know. I'd have to look. I mean, there clearly is inherent differences, probably from the parentage of these different cultivars that impose the maximum rate that can occur. So I assume it's related to the parentage, because if you take a group of species, you'll find within that group of species, within a genus, that there'll be different maximum levels, maximum PN levels that you can achieve. Does that make any sense?
Yeah.
Just related to that, what you showed were CO2 responses or photosynthetic responses that were parallel essentially throughout the temperature range that you were testing. So there must be something. It wasn't just a heat shutdown that was occurring for the resistant versus the the
Yeah, they were
less
parallel.
tolerant.
Yes.
So there must be some more fundamental difference, as you just said.
Yeah. Who is it? Question.
How about humidity, these experiments?
Yeah, with the liquor, we actually, you can set a specific humidity level and maintain the vapor pressure deficit. So you don't, you can set it so that it doesn't confound the data.
Is there any interaction between heat, temperature?
Yeah, there is. There is, yeah, very much with the vapor pressure deficit, but that actually accommodates, or the liquor actually accommodates that.
And also, did you measure ABA critical level?
No, we didn't measure ABA. The assumption is because we're seeing the guard cells closure of the stomates, that ABA is somehow involved with the process, but there's no evidence to confirming that.
And also no interaction for K plus and Ca++ deficiency or something. You didn't look at those?
No, we didn't look for nutrient deficiencies. They were treated the same, and the responses were, as you saw, almost immediate in some cases. So,
Okay.
yeah.
Another question related to the heat and shade. Oftentimes growers will say, oh, it's too hot, so I'm going to put the shade
Yeah, that's
on in the greenhouse. And so what you tell them now, okay, heat is bad, but shade is also bad, so what should they be doing?
You know, with the tomato greenhouse and the rose greenhouse, this started with the rose greenhouse. We told them to shut the shade system down. And when they shut the shade system down, guess what happened to the blind shoot percentage in the summer? It dramatically dropped. And the blind shoots they were having, they weren't having. So it solved the problem, actually.
So there wasn't that heat problem with the plants?
Well, no, let's just put it this way. The heat problem with the plants wasn't as bad as the shade problem. Okay, the shade was more limiting than high temperature reduction. Okay, did it occur? Yeah. So, and we have rose data that shows that. But it wasn't as, but the shade, where you saw, I think, just in the one example with impatiens, it dropped light levels down to nearly a third. Yeah.
What about the period of light exposure? Not low light, but high light. Because Bob Langhans, when he was here, told me that you had to put the shade over at 5 o'clock on lettuces because otherwise they had too much light. The period for photosynthesis was too long. To me, this didn't make sense, but that's what he claimed. Now, do you think that was the time for photosynthesis, or was he in fact dropping the temperature in some way? I mean, he's not here to defend himself,
No.
but have you done any experiments with the period of highlight for photosynthesis?
No. I have not. We haven't done that. And I don't see a rationale for it.
No, I didn't see a rationale, but he swore that was the case. You had to put the shade over at 5 p.m.
Where there is a stress that I think occurs on crops that we don't appreciate is actually in the morning. And you might be aware of work done with photosynthetic depression. In that case, it's actually damage to the photosynthetic apparatus when you have bright light and cool temperatures. And in England, in fact, they were able to attribute whether the corn crop made it or not was associated with how many cool, bright mornings they had. There's actual clear photosynthetic damage. It's at the University of Essex. I don't know how often that occurs in our crops. Okay, we typically don't let our crops get cold, but in some locations they do. And that actually causes suppression, long-term suppression as well. Cool and bright. When we do our temperature drop for stem elongation, I think the yellowing that we see is actually that cool temperature damage to the photosystem. Yep. Go ahead.
One of the take-home messages I got from your talk is that a greenhouse, while it's a controlled environment, it's still an imperfect place to grow plants. It's hard to, you could do a lot more to optimize carbon dioxide, optimize light, and optimize temperature. And then at the end of your talk, you showed an interesting slide with this rotating drum and plants and like that, which I think is pretty unrealistic, but maybe it's not so unrealistic to think about factory, like warehouse growing. If you could optimize all of those conditions, maybe it could be economically feasible.
Well, Bob worked on this for a while, right?
Mhm.
Bob was interested in this for quite some time. And I think it is possible to do it. It's just whether it's cost efficient to do it. Okay. The first thing I would do, frankly, and we're in the process of doing this at the university now, this room is warm and it's comfortable because the thermostat for all of us is right here. Okay? So the first thing is to take the light sensors and all the sensors associated with plant growth and put them where the plants are. Okay? Then so that's the first thing I would do as far as lesson. The other thing is I would find out where your actual light levels are, because I bet you in the growth chambers and in greenhouses you are suppressing growth more than what you appreciate. And I think that's occurring across the board, and I think it's occurring in growth chambers. As far as factory production, there are places that want to do factory production, but in many cases it frankly is a lot cheaper to just do traditional production. And I don't see the economics to do factory production yet, as long as you have accessible land. In Minneapolis, if you drive 30 minutes, you're in open farmland. You really need to spend all the money to do an intensive system in a factory when you can have a non-intensive system that may be more cost-effective 30 minutes away. I don't like to say that, but it's reality.
This is for those greenhouses with deficiency of light system within the canopy. With the future of new technology, LED light system, is it going to be solved or something? Is there a solution for that?
Well, you can create an optimal environment at the plant level. You just need to know what the parameters are that you need. The question is whether it's going to be economical, right? I mean, that's a separate question, because what I'm telling you is our plants probably could use more light than what we're giving them. At some point, it may be economical, at some point, not. So that it's going to be different. You know, frankly, we just discussed this before. In New Hampshire, the cost of electricity is 12.5 cents per kilowatt. In Minneapolis, it's 3.5 cents per kilowatt. Our economics are very different for lighting.
The CO2, you had some CO2 graphs, but I don't know if you mentioned the period or the time that was done, because there are some experiments which show that if you get a short-term burst in growth, but if you have the CO2 present all the time, there's an acclimation and it comes back to what it was before.
No,
so have you found that in your crops?
That's a very good question. The answer is no, because frankly the basis for adding CO2 to tomato greenhouses and the increase in yield is because they get a permanent increase in photosynthesis. But what we're looking at is acclimation right now. That burst in that 2-hour high temperature pulse, we're looking at if we do it periodically whether the plant doesn't depress, but actually adapts to that. So we're doing that actually right now. But I think it's a good point. Plants do acclimate. But there was actually a recent paper in Arabidopsis related to this, and they found that Arabidopsis did not acclimate. That it was just permanently increased. Do we have any more time?
Other questions for John? All right, let's thank you.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.